Good day, this is the Professor, CBMD PhD, and today's Monday, August 28th, 2023. It is 5:46 and 48 seconds, AM Eastern Standard Time, and today's research, DNA chips. So let's hop to it here. The DNA chips and storage media of the future. What challenges need to be overcome? Date August 24th. 1923. Source: University of Würzburg. In the form of DNA, nature shows how data can be stored in a space-saving and long-term manner. Bioinformatics specialists are developing DNA chips for computer technology. The scientific investigators and researchers show how a combination of molecular biology, nanotechnology, novel polymers, electronics, and automation coupled with systematic development, can make DNA data storage useful for everyday use, possible in a few years. The hereditary molecule DNA can store a great deal of information over long periods of time in a very small space. For a good 10 years, scientists have therefore been pursuing the goal of developing DNA chips for computer technology. As for example, for the long-term achieving of data, such chips will be superior to conventional silicon-based chips in terms of storage density, longevity, and sustainability. And four recurring basic building blocks are found in the DNA strand. A specific sequence of these blocks can be used to encode information, just as nature does. To build a DNA chip, the corresponding coded DNA must be synthesized and stabilized. If this works well, the information is preserved for a very long time. Researchers assume several thousand years. The information can be retrieved by automatically reading out and decoding the sequence of the four basic building blocks. The fact that digital DNA storage with a high capacity and long lifespan is feasible has been demonstrated several times in recent years. The storage costs are high, close to 400,000 US dollars per megabyte, and the information stored in the DNA can only be retrieved slowly. It takes hours or days, depending on the amount of data. These challenges must be overcome to make DNA data storage more applicable and marketable. Suitable tools for this are light-controlled enzymes and protein network design software. The research team is developing DNA chips made of semiconducting, bacterially produced nanocellulose. With a proof of concept, they can show how current electronics and computer technology can be partially replaced by molecular, biological components. In this way, sustainability, full recyclability, and high robustness, even against electromagnetic pulses or power failures, can be achieved, but also a high storage density of up to 1 billion gigabytes per gram of DNA. The researchers rate the development of DNA chips as highly relevant. They will only last a civilization in the longer term if they make the leap into this new type of sustainable computer technology, combining molecular biology with electronics, a new polymer technology. What is important for humanity is to move the circular economy in harmony with planetary boundaries and the environment. We need to achieve this in 20 to 30 years. As chip technology is an important example of this, but the sustainable technologies to produce chips without e-waste and environmental pollution are not yet mature. The nanocellulose chip concept makes a valuable contribution to this. In this new research, they critically examine the concept and advance it further with current innovations from research. The team is currently working on combining the DNA chips made of semiconducting nanocellulose even better with the designer enzymes they have developed. The enzymes also need to be further improved. In this way, they want to achieve better and better control of the DNA storage medium and be able to store even more on it, but also save costs, and thus step by step enable practical use as a storage medium in everyday life. And this is a professor of space technology, advanced physics, mathematics, computer science, and leader of research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.